Hello, my name is Professor Hudson, and you're about to view narrated PowerPoint lectures for Chapter 6 on Energy and Enzymes. How do enzymes promote biochemical reactions? All chemical reactions require an input of energy to start. This is called activation energy. Catalysts speed up chemical reactions. They speed up reactions by lowering the activation energy required for the reaction to begin. They speed up only exergonic reactions. They are not consumed or changed by the reactions they promote. Enzymes are biological catalysts. Enzymes are biological catalysts. Enzymes are employed to catalyze or speed up chemical reactions in cells by lowering the activation energy needed to start the reaction. Enzymes are biological catalysts and regulate all the reactions in living cells. This figure shows the progress of a reaction without an enzyme and with an enzyme. The red peak on the graph represents without an enzyme. The blue peak represents with an enzyme. If you notice, the amount of activation energy to make this reaction happen is less when an enzyme is present, and that's represented by the blue peak in this graph. Enzymes are proteins, and they have two attributes that set them apart from non-biological catalysts. Enzymes are very specific for the reactions they catalyze. Enzyme activity is regulated. Enzyme structures allow them to catalyze specific reactions. Each enzyme has a pocket called the active site into which one or more reactant molecules called substrates can enter. The amino acid sequence of the enzyme protein and the way the protein chains are folded create in the active site a distinctive shape and distribution of electrical charge. The distinctive shape of the active site is both complementary and specific to the substrate. Active site amino acids bind to the substrate and distort bonds to facilitate a reaction. Enzymes, like all catalysts, lower activation energy. The breakdown or synthesis of a molecule within a cell usually occurs in many small steps, each catalyzed by a different enzyme. Each of the enzymes lowers the activation energy for its particular reaction, allowing the reaction to occur readily at body temperature. There are 3 steps of an enzyme catalyst in terms of how it works to catalyze a reaction. Both the shape and the charge of the active site allow substrates to enter the enzyme only in specific orientations. Therefore, the enzyme will only bind to specific substrates. Upon binding, the substrates in active site change shape to promote a reaction. We call this the induced fit model, where the binding of the substrate causes the enzyme to change its shape such that the reaction is facilitated. When the reaction between the substrate is finished, the products no longer properly fit into the active site and diffuse away. The enzyme reverts back to its original configuration and is ready to accept more substrates. Enzymes play a role in both catabolic and anabolic reactions. Catabolism and anabolism are the 2 phases of metabolism. So when we talk about metabolism, we talk about not just how quickly somebody can break down the foods they eat, but we're also talking about how the person's body synthesizes macromolecules. Catabolism is the breakdown or the hydrolysis of macromolecules. For example, proteins breaking down the amino acids and starch breaking down the glucose. Catabolic reactions, where molecules are broken down, are often considered exergonic reactions with a release of energy. Anabolism is the synthesis, condensation, or the production of macromolecules. For example, amino acids reacting to make proteins and glucose reacting to make starch. These reactions are anabolic and often require an input of energy, and this makes them endergonic. Oftentimes, the breakdown of larger molecules with the release of energy is coupled to the synthesis of molecules. So this would be considered a coupled reaction. So catabolism is often coupled with anabolism. Let's take a look at our last topic, which are how enzymes are regulated. The sum of all of the chemical reactions inside a cell is called metabolism, and this will consist often of catabolic and anabolic reactions. Many cellular reactions are linked through metabolic pathways. In a metabolic pathway, an initial reactant molecule is modified by an enzyme, creating a slightly different intermediate molecule, which is modified by another enzyme, and so on until a final product is produced. In this metabolic pathway, we have the initial reactant A converted by enzyme 1 into B. And then this product B becomes the reactant for enzyme 2 to make C. And then the product C can become the reactant to make D with enzyme 3 or F with enzyme 5, and so on. And then our final end products in this pathway would be product E and G. With metabolic pathways, if we remove one of the initial reactants or one of the initial enzymes, you will not get any of the intermediates or end products. Let's take a look at another review question. Enzymes slow down chemical reactions, increase the energy of activation, bring together specific molecules and cause them to react with each other, or all of these. Choose the correct answer. In talking about how the active site fits together with the substrate, we used to think it was called the lock and key model, where the substrate had to be a perfect fit for the active site for the reaction to occur. However, we now know that the best description of how they interact is called the induced fit model. This is where the substrate itself causes the active site to conform around it. So instead of being a perfect fit, you could say that the substrate maybe is more of like a 98% fit. It has to be the right substrate, but then if it is the right substrate, it will cause the enzyme to close around it for the reaction to occur. As I mentioned previously, enzymes can play a role in degradation, where a substrate is broken down into products, smaller products, which would be exergonic. Or you can have reactions where the enzyme has the ability to allow smaller substrates to react to make a larger product, and we call that synthesis, which is endergonic. Let's talk about some factors that affect enzymatic activity. There have to be a way to regulate enzymes so they're not on all the time when they're not needed. One way of reversibly inhibiting enzymes is competitive or non-competitive inhibition. This is where a molecule binds to an enzyme to decrease its activity. You also have allosteric regulation, where small regulatory molecules bind to the enzyme to increase or decrease its activity. Then we also have feedback inhibition. This is where the end product of a pathway will shut off the first enzyme in the pathway and thus shut off the entire pathway. This slide shows a normal enzymatic reaction, where the substrate binds to the active site of the enzyme in order to initiate a reaction. In competitive inhibition, you will have an inhibitor molecule bind to the active site and then block the entry of the substrate. Because this inhibitor is not the actual correct substrate for the enzyme, no reaction occurs. However, because it's sitting in the spot where the correct substrate would like to bind, it cannot come in to start the reaction. We call this competitive inhibition because the inhibitor is competing with the substrate for the active site. In non-competitive inhibition, a molecule can bind to a site on the enzyme distinct from the active site. The active site changes shape so the substrate no longer fits with when a competitive inhibitor binds the enzyme. So in this case, the inhibitor binds to another site on the enzyme, and so it doesn't block the active site. However, when it binds to this other site, the enzyme changes shape such that its active site no longer will fit the correct substrate. In allosteric regulation, you have small regulatory molecules that can bind to the enzyme and enhance or inhibit its activity. And that is depicted in this slide. Feedback inhibition is a negative feedback type of allosteric inhibition that causes a metabolic pathway to stop producing its product when quantities reach an optimum level. An enzyme near the beginning of a metabolic pathway is allosterically inhibited by the end product of the pathway. So in this figure, once enough isoleucine is made, it will then go and bind to the very first enzyme in this process, shutting it off. And with that very first enzyme shut off, then you will not get the rest of the substrates necessary for the other enzymes. And so the pathway is off. In nature, there does exist items that can irreversibly shut off enzymes, oftentimes with deleterious effects on the organism. Poisons, drugs, and environmental conditions can influence enzyme activity. Drugs and poisons often inhibit enzymes by competing with the natural substrate for the active site. This process occurs either by competitive or by non-competitive inhibition. Environmental conditions can denature enzymes, distorting the three-dimensional structure crucial for their function. Some poisons and drugs are competitive or non-competitive inhibitors of enzymes. Competitive inhibitors of enzymes include some nerve gases and insecticides, which permanently block the active site. Some examples would be cyanide, which is deadly to most living organisms, sarin nerve gas, penicillin, which is deadly to bacteria, heavy metals, and other nerve gases. All of these are considered poisons which irreversibly shut off key enzymes, and without those key enzymes working, biological processes within an organism will stop and they will become very ill or even die. Lastly, let's talk about some other factors that affect enzyme activity and the rate at which reactions occur. Substrate concentration, enzyme concentration, salt concentration, temperature, and pH can all affect enzymes and the rate of reactions. Let's take a look at these in more detail. With respect to substrate concentration, the more substrate you add to a reaction, the more the reaction or the faster the enzymes will work. Of course, this only goes up to a point. At some point, you will have reached the point of saturation where all of the enzymes are busy, and so the reaction will not increase any faster. Also, you have enzyme concentration. The more enzymes you add to a reaction, the faster the reaction will go, and it will just go continuously faster. Enzymes are sensitive to temperature. Every enzyme has an optimal temperature for which it works, and if you are lower than or greater than that temperature, it will not work as well. And at certain temperatures, enzymes will lose their structure and become denatured, and then they will stop functioning. And also, most enzymes have a certain pH for which they work best at. And if you're below or above that, they will not function and can even become denatured. This concludes our narrated PowerPoint